Our speaker for today is Dr. Robert Boyd. Dr. Robert Boyd is a naturopathic physician and certified nutrition specialist, professional specializing in thyroid disorders, autoimmune disease, infertility, and men's health. He has spent years studying the fields of nutrigenomics as well as the gut microbiome and tailors many of his treatment approaches around these fascinating fields. And at this time, I would like to welcome Dr. Boyd.
Thank you so much. Thank you for the introduction. I'm excited to be here today. This is an area that I'm, really excited to talk about and share a lot of information about. Clinical nutrigenomics is something that I learned about a lot during naturopathic medical school and during my years of residency with Dr. Peter D'Adamo, and get to use it now with my own patients. And it's something that I feel is a really relevant tool, especially in today's climate. And really gives people, a new set of answers and a new set of ways to approach problems. So without further ado, we'll go ahead and get started. Just a quick outline for today. So we'll start with just a quick context and overview of what genomics is, what genetics is, just some boring definitions just to make sure we're all on the same page. After that, we'll get started with Opus 23 Explorer, what exactly that tool is, what sort of sets it apart from other genomic analysis tools and platforms out there, and really how it approaches, this interesting sort of information. Next, we'll talk about two different ways to use the program. I like to call them basic and advanced use, but they're both suited for different sort of goals and games, and so we'll talk about how exactly we use those and when to use them. Then we'll get into a couple of examples about how to approach chief complaints and, family history and social history in terms of genomics. And then lastly, we'll go over a live curation of how to actually, take some information from an intake, a couple of chief complaints, some family history, and then actually put together a full report for a client. So starting out with just some important distinctions. So I always like to start with this so that we're all on the same page. So genetics is what you learned about probably during medical school or in your training in biology and things like that. So this is the study of hereditary and inherited characteristics. So we're talking about more congenital genetic diseases such as cystic fibrosis, sickle cell, Huntington's, things like that. These are more serious in nature and are related to, either full chromosomal deletions or, really complex genetic disorders. Then we get into the field of genomics, which is we're looking at things from much more functional nature. This is how, how genes behave in their interactions with each other in the environment. And so some of the big hitters in this world are, for probably everyone who's tuning in today, they've all heard of MTHFR to some extent, ApoE, BRCA polymorphisms, as well as the field of nutrigenomics, pharmacogenomics, and epistasis. And nutrigenomics, for example, is the study of the interaction between nutrition and genes and how they interplay for both prevention and treatment. But then we also have pharmacogenomics, so that's how certain medications react to people depending on their genes, as well as epistasis, which is actually how genes interact to turn one another off or on. So, and when we're talking about this field, I always want to make sure that we start with this caveat. And, genomics, nutrigenomics, this is a blossoming field with a ton of information already available, but we're really at the tip of the iceberg. And every day new research is coming out, and every day we're learning more and more. And I just always like to say, this is just one piece of the puzzle as in comparison to everything else you know about your client or your patient. So, we always want to keep in mind what the client's actually coming to you for, what the patient's coming to you for. In the early days of analyzing this information using Opus, which is the program we're going to talk about today, it's easy to kind of get lost in the woods and put together a report that addresses a lot of different factors that the person, they really just came in to talk to you about migraines. And so we always want to keep their chief complaint in mind and make sure that we're centering everything around that and making sure that we're developing a program and a report for the person that is based on their needs. We also always want to consider their personal medical history. So there's a lot that happens in between when your genes were laid out, right? So at the moment of conception to where they are today. There's years of medical conditions they might have had. There are years of, a whole onslaught of factors that really factor into these things. And we also want to pay attention to family history, social history, diet and lifestyle factors, as well as any medications and supplements they're on. So the genomics is an important piece of the puzzle, no doubt, but we also want to keep in mind all these other aspects. So let's get into some more of the specifics now. So what exactly is Opus 23 Explorer? Opus 23 Explorer is a tool to analyze data from the DSL Genomic Insight test. This test is a SNP test, and so it's testing for thousands of different SNPs that are possible in your patients. And then it comes back and we're looking at exactly how these SNPs make someone differ from another. And then Opus 23 Explorer is the tool that we use to analyze those SNPs. So all of the data from the DSL Genomic Insight test gets funneled into Opus 23 Explorer, which allows a trained practitioner, a doctor, anyone who can order that test to really analyze the data and provide a very in-depth report for their patient. Opus 23 Explorer is actually, so it's kind of, it's rolled together as one program, but it's actually multiple different analytics applications rolled into a single program. And when we go through the live demo later, we'll see how there's different ways that we can sort of slice the data and look at things from a different perspective to help generate the relevant information. Very importantly, the Opus 23 uses visual informatics to present what can be pretty complex information in a very user-friendly way. So we, this field has a fairly high barrier to entry, right? Genomics can seem overwhelming and can really seem like there's a ton of information out there, and where do you get started? Opus tries to really take the guesswork out of it for you and present the information in a way where it's always drawing your attention to the most important areas. So really, if you're paying attention and you're going through the program, you're always going to end up finding some of the more relevant information for your client. And then also very importantly, all data within Opus 23 Explorer is sourced from peer-reviewed science-based articles and sources. So there's nothing within the program that doesn't have some sort of PubMed citation attached to it, or some sort of ClinVar database, or one of the other SNPpedia database attached to it. So we're always pulling from sources that are reputable and have high quality information. And then last but certainly not least, this is a new tool to engage your patients and clients. A lot of people are coming to their doctors these days with their file from 23andMe or Ancestry or wherever, and they're wanting these things analyzed. This gives you an opportunity to say, hey, let's take a look. This could be another piece of the puzzle, as I said earlier, and we'll see if we can get some answers from the more genomic side of information. And within Opus 23, you're able to provide customizable client reports, so you can provide as many reports as you want to your client. You can also provide information on pharmaceuticals and pharmaceutical and etiological data. So you're able to see exactly how their SNPs are interacting with medications they might be on, or medications they might potentially go on. So we can, get in front of drug interactions and how, side effects that people experience. And then etiological data as well. So when we say etiological data, we're looking at potential factors that are contributory to, things that they should avoid in order to, avoid making certain health conditions worse. And then lastly, within those reports, you can also customize supplement, dietary, and lifestyle recommendations. So we can get really specific in terms of, which specific herb or nutrient or vitamin we want to recommend someone, specific lifestyle factors and dietary choices as well. And one of the amazing things about the reports too is that whenever you pick out a specific gene or a specific SNP to focus on with someone, it always ties in, okay, this is why, Dr. such and such is recommending magnesium. It's because of this effect on this gene. And so a lot of times it takes the guesswork out of people for, why they were recommended one supplement or one lifestyle change over another. So in terms of approaching the program, so there's two ways we can go about it. The first way, and I'm going to talk a little bit at higher level about these things to start, and then during the last portion of this presentation, we're actually going to go into a live demo so you can actually see these things. So the first way that we address these things is, one of them is the report wizard. So the report wizard is, I say quote unquote basic use because there's nothing basic or simple about it. It's pulling together this amazing amount of information all at once. But it's more for if you're wanting to generate a quick report for someone based on kind of a general area. So it's not going to give you as much flexibility in terms of picking out specific genes or picking out specific algorithms or agents or things like that, but it allows you to develop a quick report about inflammation, for example, or a quick report about neurodegenerative disease, or a quick report about hormonal control. The second way to approach Opus 23 Explorer is through the curation editor. So this is the quote-unquote advanced use of Opus 23 Explorer, where we can actually fully customize the report, down to the very last gene or our algorithm or all these different things where we can actually pick out exactly what we want to go in there. Obviously, this one, it does take a little bit more time compared to the report wizard where it's, a couple of quick clicks and you're done. However, the curation editor, even though it takes more time, it doesn't take an extraordinary amount of time, and it gives you a high amount of flexibility in terms of what you're going to be discussing with your client. So starting with the report wizard. So as I said, it's more of a fundamental use, a lot of quick general report curations. You can actually choose up to 3 categories to focus on for a report. And after you choose those 3 categories, so you can choose from 1 to 3, it generates a report with the most important relevant genes, algorithms, and agents. And so just to give a quick photo of what that actually looks like. So, and we'll see this during the live demo as well. The report wizard, so you can see all these different categories here. So we have methylation, detoxification, cognition, inflammation. I won't read them all, but you see that some of them have been selected. So for example, detox, inflammation, and then at the bottom, androgen genomics. And so what we're choosing right now is to create a report based on those 3 things. So what Opus will do is it'll crawl through all of their genetic information, genomic information, and pick out the most relevant genes in each of those categories where there are SNPs that are showing to have a high effect. You can also choose, and you can see out to the right here, you can choose to include the network maps. So when we say network maps, we're talking about the different biochemical and genetic maps that contribute to these areas. So for example, the network maps for detoxification will lay out the phase 1 cytochromes, phase 2 glutathione detoxification, and sulfation and all those good things. And then you can also choose to include introductory explanatory text and a couple of other things here. During the live demo, we'll cover the report wizard briefly. From my perspective, I always want to use this second version of the quote-unquote advanced use, because it allows the flexibility of actually picking out the specific genes in the specific areas you want to focus on. When it comes to trying to create a report for people based on based on their chief complaint or what they're coming to you for, we want to be able to provide that level of customizability, and that's what this does. You can, and also, an important part of this here is that we can actually develop multiple reports in different areas. So let's say you have someone, you have a client that's a complicated case, right? They've got a lot of different things going on. Well, let's say they have, migraines and anxiety and Crohn's disease. We can develop individual reports based on all those areas. So you can say, okay, so this week we're going to focus on the Crohn's. We're going to look at some of the inflammatory markers associated with that. We're going to look and see if there was any sort of genomic susceptibility there, and then try to put around something in place around that. So a couple of therapeutics and a couple of areas to address there. And then in 2 weeks, we'll come back and we'll look at the migraines, and we'll put together a report about that. And so we can always, this allows you the flexibility to really choose which specific things you want to address with people. And, big picture things I always stress for people when we're using the more advanced version of curating, which is using that curation editor, is we always want to keep the chief complaint in mind, which I said earlier. very importantly, and I'll show this during the live demo, all of the applications within Opus, so whether or not you're looking at the network maps, or you're looking at individual genes, or you're looking at individual SNPs, all of those feed into the curation editor. So anytime you choose something within the program, you don't have to worry about keeping track of it yourself. It will automatically feed it right into that editor. And then at the very end, you're able to pick and choose what exactly you want to go into the report out of those things that you chose. And then it's ready to go, and it automatically generates it. And then lastly, Opus 23 Explorer is a great teacher. So even if you're not an expert in the field of genomics, which I think there are a few people on this planet that can truly call themselves that, I'm not putting myself in that category at all. The more and more you interact with this type of data, the more you'll learn. And so Opus 23 does a great job of always, anytime you click on a gene, for example, you are always getting to read about what that gene and enzyme does as a function. So if you see that, 5 times, 10 times, every time you click on it, things start sticking. And so the more and more you interact with the program, the more and more you actually pull from it and begin to enrich your own information. And so the first time you open up the Opus 23 Explorer and you're clicking around and you see the different applications, you might feel overwhelmed, but rest assured that everyone feels that way, and that as soon as you get an understanding of how the program works, which is what I hope to achieve today, the more and more you interact with it, the more that you're actually going to see that it's actually teaching you and informing your decisions much more than you would expect. So now we're going to dig into a couple of different applications. We'll cover all these during the live demo also, but I wanted to show them in sort of this flat mode so that we can be exposed to them before all the moving parts get into play. And remember from the previous slide, I mentioned that all the applications, so the next, I think there's 4 or 5 here that we're going to talk about, these all feed into that curation editor. So when you're within these applications, and for example, if you click that you wanted to curate this gene right here, it would automatically feed it into the curation editor, and the editor is where you go at the very end to generate a report for a client. So all of these things, the curation editor is what sort of acts as like a catch-all funnel. And so anytime you're clicking around and you say, oh, this is interesting, I want to save that or this or that, it all catches it, and then at the very end you can pick and choose exactly what you want to go in that report. The SNP navigator does exactly that, so it navigates the SNPs. You'll see up here on the top right where it has a little search section. This is actually an active search bar where you can type in a word. So for example, I typed in Alzheimer and then took a screenshot of this. And so it's going to pull up every gene and SNP that has the word Alzheimer in the keywords. You might not see it in some of these ones at the top because it's in the entire list of all the keywords, but rest assured it's in there. And then you can see it's actually sorting it by the relative magnitude. So how important this actual SNP is in relation to the function of the gene. And so you can start to, if you've got someone coming to you with, a family history of Alzheimer's, or they've been currently diagnosed with Alzheimer's, you can come in here and look for some of the more relevant SNPs. So for example, this second one on the list, TCN2, they have a homozygous risk allele for that. So it's possibly somewhere to pay attention to. I always say that if you don't know really where to begin with this, with investigating the applications, it's good to come here because you can literally just type in the patient's chief complaint and go from there and let the program take you. And so you can really get an idea through a very highly flexible search here. The next application we're going to talk about are the network maps. So these network maps are actually genetic maps on how different genes and gene products feed into each other. So you'll see here there's 10 of them within Opus 23 Explorer, and then you can see the actual different categories that they're tagged for. So when they're related to inflammation or gene expression, detox, endocrine, all these different categories. And so if you've got someone, and when we get into the more specific areas about how to address an actual problem, you can sort of lump things into these categories and then figure out which map you want to go with from there. So let's pretend like I clicked on one of these maps. We'll say that I clicked on the 1-carbon metabolism, and then this is what comes up. So now we're actually looking at the 1-carbon metabolism pathway. What I love about this compared to some of the other genomic analysis programs out there is that this is actually an active representation of the client's genome. And so where you see orange bars on the genes here, so you can see all the different gene names, for example, MTHFR is down here, TCN2, all of your MTR, MTRRs here. Where you see orange bars, that's actually where they have homozygous SNPs present. So you can actually get a quick idea of where there's actually compromise in this pathway. When we see yellow bars, this is actually heterozygous mutations. So one risk allele, one normal allele. And then lastly, gray means normal wild type. So they didn't have any mutations there. And so when we're looking at this map, we can get a quick idea of based on how full those bars are, where potential hangups are in this pathway. Another application we have is called syndromes. Syndromes actually takes those same genes, and then instead of grouping them onto biochemical maps or listing them by their SNPs, it groups them into possible syndromes that they're related to. So for example, the two that I have listed here are thyroid genomics and adipocytokine genomics. So, this is more of inflammation related to metabolic diseases. This is grouping on a sort of this agent fingerprint pattern, what exactly, what genes are involved. So for example, we have VDR for the thyroid genomics, DIO2 and DIO1 over here, which are the enzymes involved in converting your thyroid hormone from T4 to T3 and so on. And so we're able to take those, and this is what I meant when we can slice the data in a lot of different ways, is we can take those same genes we've been talking about all the time and put them into categories, so that if you've got someone coming to you with hypothyroid, for example, you can immediately come over to this thyroid genomics area and look at where we're experiencing any sort of possible homozygous or heterozygous mutations. And just a quick note, anytime throughout the program, so anytime you see yellow, that's representing heterozygous mutations. And anytime you see orange, that's representing homozygous mutations. So we keep the coloring throughout the program consistent so that it sort of kind of trains your eye as you go, so you get used to all the different schemes and patterns. One of our next applications is called algorithms. So what an algorithm does is it takes either multiple genes or multiple SNPs and then fits them into a clinical scenario. So what do I mean by that? So there's types of studies out there called GWAS studies, so genome-wide association studies. And what they'll look at is okay, so this cohort of population had this, these SNPs in common. Okay, so which diseases do these people have in common? And so they're able to discover SNP associations with certain diseases, or risks for certain things. So for example, increased risk of chemobrain, CRP protein genotypes and how it relates to inflammation, increased risk of metabolic syndrome. So these are all algorithms that this individual that we're looking at their genomics for was true for. And so there's over 100 of these within the Opus 23 Explorer, and they range in a wide amount of categories in which they relate to. So you can see out here on the left, this one up here is related to oncology, whereas the next one's related to autoimmune. We have pharmacogenomics, neuropsych, endocrine, cardiology, and you can once again come within the program and search into this search bar and actually get you can type in, for example, cardiology if you want to see only the cardiology algorithms. And then last but certainly not least, we get into sort of the less of the informational side and more of the action side. And so we get to the pharmacogenomics, for example. So pharmacogenomics being how different pharmaceuticals interact with people based on their SNPs. Certain side effects that you can look out for, or possibly explain certain side effects, or actually you can actually use some of this information to actually direct drug therapy if you are a prescribing doctor. The natural products, so we can actually look at which natural products are most well indicated. And then lastly, the Logos AI prescriptive tool, which actually ranks the most compromised genes for you in sort of a tabular pattern and then recommends whether they should be slightly upregulated or downregulated. So in relation to using different herbs. So for example, if we want to increase the activity of MTHFR because someone has double homozygous mutations, it would recommend methylfolate to do that activity. And so we can actually begin to use the program to actually direct specific recommendations, whether they're pharmaceuticals or natural products, as well as there's also algorithms and a completely different subset of the program for recommending and managing designs for health products, which is tied into the program as well. Or you can completely choose just natural products separate from designs for health products. And so let's get into kind of a more specific how we want to actually approach the problem. What I've found to be most helpful for people when we were, when I was working with Dr. Diadamo, who was, the creator of this program, as well as and working with naturopathic medical students, and nowadays in my own practice, we want to group things, right? So we want to look at how, what someone's coming in presenting with, and then try to figure out which sort of these groupings of conditions they fall into. And then once we figure out where these, which sort of categories, for lack of a better term, they fall into, then we can let this sort of category search direct our approach throughout the program. So for example, and these are like the sort of the categories that I use in my mind and try to lump things into. We can lump things into a cardiometabolic category. So these are people coming in with cardiovascular disease, diabetes, overweight, obesity, detoxification issues. So if someone's coming in with a lot of hypersensitivities, hormonal imbalances, fatigue, we want to look down their detox pathways to see if those are affected. Cognition. So we see if there's a family history or a current medical history of any of these sort of neurodegenerative disorders. Endocrine being, if they've got thyroid disease, adrenals, sex hormone imbalance, as well as insulin insensitivity or insulin receptor issues. Histamine. So when I think of histamine, we're going to think of allergies and sensitivities, individuals who are hyperreactive, as well as a lot of ADD and ADHD actually plays into this histamine picture as well. Immune dysregulation. So we've got autoimmune diseases, IBD, so that includes Crohn's, ulcerative colitis, chronic infections, allergies and sensitivities. Inflammation, so we have autoimmune disease, rheumatologic diseases, some of the other things that we talked about. And then lastly, these three categories, female and male hormones. So I listed out some common female conditions as well as male. Methylation, so I put everything here as kind of a joke, but kind of not, because as a lot of us know that methylation has a lot to do with everything. It's hormonal detoxification, neurotransmitter recycling, preventing DNA damage. I mean, all of these different things are so important and critical for a lot of different conditions. So we always want to pay attention there. And then lastly, their neurotransmitter metabolism. So we have ADD, ADHD, anxiety, depression, elevated fight or flight response. And so when I get someone coming in and we want to take this sort of genomic approach, and what I do is I think about these categories. And I think about with what the conditions they have, what areas do I need to focus on when I'm trying to generate a report. So, if someone, for example, is coming in and they have, let's see, like a family history and a personal history of cardiovascular disease, sure, a lot of these different categories can play into that, but there's also specific areas where I definitely want to make sure I look at. So, for example, I want to check and make sure that they don't have any of the genes and SNPs associated with cardiometabolic diseases. I want to check and make sure inflammation isn't involved. So we know that a lot of diabetes and things like that, these are inflammation-based diseases as well. And then lastly, methylation. And so, on these next couple of slides, what I wanted to do is go through a couple of kind of quick cases in terms of, what areas we would want to investigate, and then spend the last 15, 20 minutes or so actually going through the live curation of how we would do one of those cases with an Opus. And so example number one, and I put this at the top of every one of the examples, we want to look for common threads in their history and then investigate those areas. And you'll see over here on the right side, I listed out these sort of quote-unquote genomic categories where we want to actually make sure we're investigating these areas. The base of them are all in blue, and then the ones that I would immediately jump to as most pertinent, I've highlighted in green. So let's go through the case. So we've got a 28-year-old female presenting with concerns over cyclical acne. So immediately we're thinking, okay, so it's related to their hormonal cycle. Okay. She also has a history significant for uterine fibroids and a family history significant for breast cancer and endometriosis. So immediately, what we're wanting to look at is the sort of female hormonal cascade, right? In terms of their estrogen metabolism, their estrogen detoxification, and the receptor signaling. And then we also want to make sure their genital detoxification pathways are functioning well, because those are also involved in a lot of the hormonal detoxification. And so some of the main genes I would, immediately think to, and these are listed together within Opus, are ESR1 and 2. So those are estrogen receptors 1 and 2. The CYPs, so cytochrome P450, the ones that are involved in the 2,4-hydroxylation of estrogen, as well as aromatase. Sulfation enzymes, sulfation genes. So sulfation is also a big part in terms of detoxifying estrogens appropriately, as well as glucuronidation. Thinking back to when we learned about phase 2 metabolism back in medical school or naturopathic medical school, or wherever you learned about it, we think about glutathione, we think about sulfation, glucuronidation, all these different things. And then lastly, we have methylation of the estrogen metabolites. So making sure that they don't end up accumulating and causing accumulation of DNA damage. And so when I see someone like this, for example, within Opus, and I'll show you during the live curation, we can actually begin to direct our approach immediately to focus on these two areas. And sure, eventually we might want to look for in this person if they have APOE, which is related to onset of Alzheimer's disease. But obviously, this person's coming to us over these cyclical acne issues and the uterine fibroids, and in the family history, we see the breast cancer and endometriosis, which immediately points us more into this hormonal cascade. So while there's important areas, surely, in all of these different sort of categories here, we want to make sure that we're doing the client a good service and not, taking them down the rabbit hole of all these different pathways, when really what they came to us was this acne chief complaint. Example 2. And so once again, we want to look for the common threads, right? So we've got a 39-year-old male coming in with concerns of overweight. His past medical history is significant for prediabetes, so maybe he has a hemoglobin A1C that's creeping up, maybe in the 5.8 range, 5.9 range. He's got a family history significant for multiple cardiovascular events. So we automatically, and these are kind of simplistic examples. I know that no one comes in with this clear cut of a picture, but I just wanted to illustrate it for the purpose of the genomics here. Automatically, we're going to end up thinking about the cardiometabolic aspects and the inflammation aspects. And so within those two categories, and you'll also see that these categories here line up with a lot of the sort of how the network maps interact. And so I'll show that as well when we get to the demo. So for the sort of adipocytokine signaling and metabolic signaling, we want to pay attention to genes like AdipoQ, which is adiponectin, which is an anti-inflammatory hormone that's inversely associated with weight and overweight. So if someone has high overweight and difficulties with insulin signaling, they're going to have lower levels of this good anti-inflammatory hormone. We also want to look at their insulin receptor signaling. So it's not only necessarily that they're becoming insensitive to insulin, but also their receptor can be a lower signaler compared to someone else's receptor. So that might also be a predisposer to these prediabetes and overweight concerns. PPAR gamma. So we have PPAR-G, which regulates adipocyte differentiation in the cell. And then lastly, leptin receptor resistance, which is becoming more and more of a well-known area of controlling overweight and obesity. And then lastly, with inflammation, we can actually look at the genomic function of CRP, NF-kappa beta 1, which these are two major players in terms of inflammation. And then lastly, ICAM and VCAM, which these are actually signaling molecules on the lining of the endothelium of the arteries, to begin leukocyte adhesion. And so we can look at their susceptibility to developing plaques in their arteries and things like that. And then lastly, this is our sort of last example, and we're going to take this example and sort of run with it within the program. So we have a 41-year-old female coming in with concerns of anxiety. She has a past medical history significant for high blood pressure, for which she currently takes metoprolol. And then so when we see someone coming in with these things together, we're going to immediately think about methylation, neurotransmitter metabolism, and you could even, make an argument in regards to the high blood pressure to look at the cardiometabolic, but for our sakes here, we're going to just stick with the methylation and the neurotransmitters. And so within the sort of genomic categories and genomic areas to investigate, what we want to look first at is, catecholamine metabolism. So, for example, if someone is writing with high amounts of catecholamines, so norepinephrine, epinephrine, they're going to, be at a higher risk for things like anxiety and high blood pressure. So, some of the genes we would want to investigate in that area are DBH. So this is dopamine beta hydroxylase, which converts dopamine into norepi, or noradrenaline, depending on which area of the world you're from. And then also COMT and MAOA/B. So those are the two that are involved in the actual metabolism of norepi and epi. And then lastly, for the methylation genomics, this is a very important part here because, a lot of times with genomic reports out there, they're really just looking at MTHFR, whether or not someone's homozygous or heterozygous for the C677T and A1298C SNPs. There is much more to that pathway than just those two SNPs and that gene alone. And so we want to pay attention to that entire thing. And so let me go ahead and switch it over to my Chrome browser. Give me just 1 second here. Okay. So we should be able to see my Chrome browser now. I started on this page for a very strategic reason in that this is the report wizard. So this is probably not what we're going to talk about today. This is the more simplified use of Opus that is much more easily looked at on your own, and you can just kind of play with it after you're using the program. This is the one that allows you to sort of pick 3 different areas and then generate a report based on it. So you can run the wizard down here, and it will generate a report of the most common genes, SNPs, network maps, and as well as pharmacogenomics and most relevant agents. And so you can choose to do one at a time, which is what I would recommend if you're just starting to use the program. So if you want to just use it to generate a report on detoxification, then you could just click that one and then come down and run the report. Or if you're feeling up to it, you can choose 2 or 3 of these categories. Where I want to focus our efforts for today during the demo is this down here called the curation editor. Now, I'm gonna show you this now, and it's completely blank. So, for example, we have no genes selected, we have no templates selected, no multi-SNP macros, which is also called those algorithms, no maps, none of it, right? So it's completely different. It's completely not different, completely blank. And so the reason I'm showing you that it's blank is that as we begin using the different data management applications over here, you're going to see that these categories begin to fill up, okay? And after we've gone through all these different applications, we'll actually be able to come back here to the curation editor and see that it kind of caught everything like a funnel, like I mentioned earlier, and then you can choose exactly what you want to go into this generate client report, and then that's it, and it's ready to go. So starting with the SNP navigator, and I'll just go through each one of these in a row, and just to orient you, so this is the main login page of Opus 23 Explorer. At the top, you see the name of the client that I have loaded. Her name is Venus DeMilo. This is a dummy data, so we're going to call her Venus. So we have her loaded. If I wanted to, this is an administrative tab, so you typically wouldn't see that one in your login. The next one down is called patient management. So you would click on this to get to all of your other patients, all of your other clients. So right now we have Venus loaded, but let's say I wanted to upload John Doe's data. I could come here and list load his information. The next 5 over here are our main 5 sort of categories of applications. We talked about each one of these during the presentation portion. So we're going to start with the SNP Navigator. So I come here and I click this. And what we get to is our main sort of page for the SNP Navigator. You can see that it's sorting here based on magnitude, and you can see that the bar here is grade. So the highest ones are 10, they're listed at the top, and then it goes down all the way to 1. We can come in here and we can search. Now, I always tell people that if you don't really know where to start, but you're wanting to create these sort of custom reports, to start here, because you can literally type anything into the search bar and then let it take you from there. So for example, with this individual, we've mentioned anxiety, right? So you can see right here, right at the top, COMT is one of the first ones that pops up. You can actually click on this gene, and when you click on it, what we call an agent pop-up, or a gene pop-up appears. How a gene pop-up is structured is the exact same for every gene within this program. So the very first part is an explanation of what exactly this gene or this enzyme does. Okay, so this one is a relatively long. However, if I thought this gene was important for our client here, and I added it to the report, the Opus does a great job of explaining things to people, but you don't want a patient to have to read this and for them to be completely confused, because it's just a huge explanation for something. So what it does is it actually puts together a much shorter client description, so that, and this is written in terms so that someone who hasn't taken a genetic class in the last 5 years can actually understand it. And so this is actually the explanation that would go into the report. So we can scroll down, and we also see the references for that entire long explanation, all the algorithms, so we call them macros, that COMT is involved in. And I'll go through those when we're actually looking at the macro screen. All of the relevant SNPs. So this is generally what individuals, if you've ever uploaded your genetic information into another sort of genetic analysis program, this is pretty much all they give you, maybe with a little explanation out here. So they just give you kind of this little tabular layout with all of the SNPs, and then they might tell you, they'll color it red or yellow or whatever, and then that's pretty much it. So we include this because certain people do care about the individual SNPs, and so we want to show those. And then last but not least, and really one of the most important aspects here, is the agent fingerprint pattern. So these are all of the agents that we're currently tracking that have been shown to either have an agonistic effect, so an upregulating effect on COMT, or a downregulating effect on COMT, or an antagonistic. So green being upregulating, orange being downregulating. And so you can see here, for example, that magnesium is a very strong agonist, whereas cannabis sativa is actually a strong antagonist. For someone who's coming in with a complaint of anxiety, we would probably want to upregulate the activity of COMT, because it's going to actually help with getting rid of a buildup of norepi and epi if that's going on. And so if we wanted to, and let's pretend like we've gone through a lot of the genomics here and we think that this gene is important for this person. As soon as we click this button up here, it's going to tell us you've just added this to the curation editor. So now if I were to go back to the curation editor, COMT would be showing up there. I'm going to click back into this here, and then let's say that we think that, so we thought this gene was important. Now let's figure out a recommendation for them. So we see magnesium is a strong agonist, and also pyridoxal 5-phosphate. And we know that we want to upregulate this because it's going to help lower their levels of norepinephrine and epinephrine buildup. So we can come here, and you can actually click on this bar for magnesium, and then you can add magnesium as a curated element to the curation editor. So now it turned green, so it knows that it's in the report. Now we can come out of there. So what we've done so far is that we said that we think that COMT is an important gene. and then that we want to recommend someone to take magnesium because it helps with upregulating the activity of COMT. Let's go on to the next application. So that would be the network mappings. So here we've got all the different maps. So let's go into the catecholamine one, and you can see these categories kind of line up with the genomic categories that I spoke about earlier. So if we go into the catecholamine metabolism, we can see here the entire cascade of how L-tryptophan and L-tyrosine get turned into the metabolites VMA and HVA, and everything in between. Let's say we're looking at this, and so we already talked about COMT, so I don't want to harp on it too much. So let's say we want to pay attention to MAOA, because MAOA is also involved in the breakdown of these catecholamines. Not only would helping to reduce the amount of catecholamines, help possibly with anxiety, but also we want to reduce the blood pressure, right? And so we see MAOA, and once again, sorry, I scrolled through it. We see that there's the explanation per usual, and then we have the shortened client explanation. And then we want to pay attention to this one, so I'm going to go ahead and curate it. And then when I come back to it, now you can see that this button up here is blue. And then we see that as we come down, once again, the algorithms that it's involved in, the tabular SNP pattern here, as well as the agents. Now for MAOA, you can see that, and so once again, this is probably something that we'd want to upregulate because it's going to help with cycling out any buildup of norepi and epi. You can see here that progesterone is actually the strongest agonist that we're tracking. Well, we're not necessarily going to put someone on progesterone because they have anxiety and high blood pressure, right? That would be kind of an abnormal thing to do. So we can actually come down and we can look at other ones as well. So we see that riboflavin is actually a strong agonist of this. Lithium is, ashwagandha. Ashwagandha is also a strong agonist here. You can see the green, or not as strong as progesterone, but it's still an agonist. And so if we wanted to choose ashwagandha, and it's down here in the list, it lists them all as well. And we want to choose ashwagandha because it's a calming adaptogenic herb for them. We can come here and we can curate ashwagandha now. So now we've done two things, right? So based on this map, we chose MAOA, and earlier on the SNP navigator we chose COMT, and then we chose COMT for the magnesium content, and then MAOA with, not for the magnesium content, for magnesium's action, and then ashwagandha for its actions on MAOA. Now, let's say that you also want to share this actual map with your client so that you can actually talk through this cascade with them. So if we want to actually share this map with them, we can come up here and actually curate the entire map. Okay, so now it's going to actually print this map out in their report. So if you're a doc or a practitioner that likes to share maps and things like that and draw things out for your client, you can actually do that. Lastly, we have, and then we also have the syndrome templates. I'm not going to go into this one as depth for now because we're kind of addressing it from more of a map and SNP navigator perspective, but you could also use these categories and, if we had one, so for example, someone had neurodegenerative, we would maybe click on this. And then also for the multi-SNP macros. So this is our fourth one down the list now. So we can go to, if we wanted to first type in anxiety, for example, up here in the search bar, we can actually see that we have there's an algorithm that's true for them that says effectiveness of kava for panic and anxiety disorders. So maybe we want to share with this information with them that, hey, next time you're experiencing anxiety or you notice your blood pressure is spiking, maybe we can try a cup of kava tea or taking a kava supplement, for example. So we can curate that, okay? And now it's going to show up as a curated algorithm. So obviously we're taking a very simplistic approach for now so that I can get through everything, but we would probably spend a lot more time going through all of these different algorithms. And you can see, so I always sort them based on their importance, and then I always sort them based on true, which ones are true for them, because we want to be talking about algorithms that are actually true for them. You can actually go through these, and if there was any other ones that you thought were important for them, so let's say we think that having low levels of B12 is important, we can click that box and then come down here and save. Okay, And so we've chosen a couple of those multi-SNP macros now. Now let's go back to the curation editor. So we can see here that we've filled in some of these boxes now. So we've got some genes and SNPs in COMT and MAOA. We've got 2 of those algorithms that we chose. Lower serum levels of B12, effectiveness of kava for panic and anxiety disorders. And then we chose that we wanted to include the catecholamines in there, the catecholamine map in there. So now when I go here to generate the client report, oh, and I just want to make sure one thing real fast, that we actually included the natural products. So we chose magnesium, and we chose ashwagandha, right? So I come down here and I say those are agent curations. Now when I go back to my curation editor, we see those 2 down here, and then I hit generate report. So what we get to here is the report that you would actually send home with your client. This prints out as a nice PDF, or you can share it with them. I'm sorry, it prints out nicely from the browser, or you can share it with them as a PDF. The first section is just an explanation of what Opus 23 Explorer is, how the program assesses genetics, how it looks at things, where it gets its data, some just cookie cutter information about the program itself. I'm going to scroll through it because it's just a lot of text for now, and you can have them read through that on their own. The next section is actually the genes that we chose that we thought were most important for them. So this is just showing an example of how it's laid out. So I'm actually going to go to the one that we chose. So you can see that it lists the name of the gene. It shows how many, the orange boxes being homozygous mutations and the yellow being heterozygous. And then also any particular SNPs that are relatively important, as well as the simplified explanation here and the OWL here that actually defines words up in this explanation that they might not know the definition to. And then you can see magnesium is showing up here as this is, our clinician identified this natural product that has an beneficial expression. Same for MAOA. Once again, the simplified explanation, all of the different, it's showing how much of the gene is affected, the relevant SNPs, and then also the agent that we selected. Then as we get down to the algorithms, or the multi-SNP macros, you can see that we chose, and this first part is just an explanation of it. You can see that we chose that B12 is an important one for them, the low serum levels of B12, and then the effectiveness of kava for anxiety disorders. So literally everything that we chose when we were going through all the data just shows up in the report. And then we can see drug interactions, so whether or not there is a possible negative interaction they should be aware of. So, they might not be on any of these drugs for now, but in the future, this is good information for have. And actually, and we don't see their medication on here, metoprolol, so we know that they might not have a genomic side effect from that. So, whether it's bradycardia or, just an abnormal reaction. And then lastly, it'll also list any other natural products that are well indicated for them. And you can see that the magnesium and the ashwagandha show up down here at the very end because we individually curated those. And then also at the very end of the report, so when you print this out, you can actually include this map, which has all of the different genes on here and the SNPs and all of the relevant information. You can go on here and draw as much as you want and explain to them the entire process. And that's the entire report. And so you can see how the data within the program actually translates down to the curation editor and then back into the final report itself. Let me switch it back to the PowerPoint, and then we will go ahead and finish up. So just going down to our, so we just went through the live demo and the curation. And then, the takeaways for the presentation from itself. So we always want to keep the big picture in mind, right? So make sure that you're paying attention to their chief complaint and their medical history and trying to assess whether or not these things can kind of fall into one of these categories out here. And I always, like I was saying, I always find it's helpful to lump things together so that you don't get lost in the program and you can really focus on inflammation or you can focus on detox. And you use those categories to find those commonalities between their medical and family history. And then lastly, as I said at the beginning, this is just one piece to a very complex puzzle of a person. And so we want to, keep this information within context, but also make sure that we're paying attention to everything. And I think that's all that I've got. Thank you so much, and I hope you enjoyed the presentation. And if there's any questions, I'm happy to answer them now.
Thank you so much, Dr. Boyd. We have time for a few quick questions. Our first question is the GCH1 included in the mapping anywhere?
That is a great question, and I'm certain it is. I'm not 100% sure which map it would be, and if it's not in a mapping somewhere, then it would certainly be in the list of the SNPs and genes where you could, go in individually and assess which SNPs are affected and how much relative impact there is on that gene.
Thank you. We have another question. Is Opus 23 Explorer different from the original Opus 23? For those of us who are already certified in Opus 23 and have access to the software, how do we access this version? Is it different?
Yep, so that's a great question. So Opus 23 Explorer is completely separate from Opus 23 Pro, which is the original version of Opus. This version of Opus is only for the interpretation of the DSL Genomic Insight Test. And so if you were to order that, order the DSL Genomic Insight Test and you choose the option where you want the data imported into Opus 23 Explorer, then it will be uploaded into this. Within the context of the DSL Genomic Insight Test, you also have an option to, when you order the test, to actually just receive the raw data file of the results, and you can still take that raw data file and upload it into your Opus 23 Pro account.
Thank you again, Dr. Boyd, for a great webinar today. Thank you to all of our attendees for taking the time out of your busy day to join us. This webinar was brought to you by integrativepractitioner.com and sponsored by Diagnostic Solutions. Thank you all, and have a great rest of your day.